today. My name is Pratt, and I'm going to be giving a little bit of an intro to human microbiome research. And I'll also tell you a little bit about myself. So I grew up in Winnipeg, and I got my Bachelor's of Science in Biochemistry from the University of British Columbia in 2014. I am about to graduate with my Master's of Science in Medical Microbiology and Infectious Diseases from the University of Manitoba. And I have been working with the Public Health Agency of Canada through the National Microbiology Lab since about 2017. And my talk today has to do with my master's project. So a couple of learning objectives just to get us started and so you know what to expect from the talk today. I hope that by the end of this talk, you will be able to define and describe what a microbiome is, as well describe some techniques that are used to study microbial communities. Together, we're going to discuss some of the basis for and the application of modern microbiome research. And hopefully you will be able to have some skills so that you can recognize some of those more sensational microbiome results in advertising. And you'll see what I mean by that in a little bit. So we'll start with some definitions. So what is a microbiome? I would define a microbiome as an established microbial community inhabiting a given site. So this is a pretty basic definition, but then we can expand a little bit more on the key concepts, which I've underlined here. So a microbial community can include bacteria, viruses, archaea, fungi, and other microscopic organisms. However, I do want to point out that most research to date focuses on the bacterial component only. Next, it's going to be important that we describe what is meant by a site. So if you've ever taken a microbiology course or even attended a science fair, you probably know that you can swab just about any surface and find microorganisms. However, for microbiomes, we're talking about established microbial communities. So typically we're interested in studying communities that have co-evolved with their surrounding environment, and this helps us to learn more about that environment. So microbiomes have been isolated and described from a variety of sites, and these mainly are environmental or host sites. So we have things like soil microbiomes, ocean microbiomes. A lot of people study the microbiomes of farm animals like pigs and chickens. And then of course we have human-related microbiomes, and there are several depending on specific body sites. So we have a skin microbiome, an oral microbiome, airway, and gastrointestinal microbiome. I also want to point out that you may hear the terms microbiota and microbiome used interchangeably when it comes to microbiome research. There is a slight difference. So the microbiota actually refers to the microbes themselves, while the term microbiome is supposed to describe the community as a whole. So this includes the microbiota, the organisms, and all of their genetic material or genomes. And this is to tell us more about the functionality of that community. So let's say if all of us in this virtual classroom were part of a microbiome, I could collect everyone's name, and that might tell me about the microbiota. But in order to understand our group as a whole functioning community, I would need to gather information about what everyone does. So maybe I would ask you what your area of study is or what your hobbies are, and that would be our microbiome. So I want to do a quick little activity just to get us started. So I'm going to ask you to head over to Mentimeter, and I'd like you to tell me, what do you already know about microbiomes? What kind of preconceived notions do you have? This doesn't have to be in the context of school. So this could just be, if you've seen advertising, if you've heard something on TV or whatever. If you don't know anything, it would be helpful if you told me that as well. So head on over to Mentimeter, and we can start. I'm just going to use your answers to inform the rest of the presentation, how much time I spend on things. You can also feel free to unmute yourself and we can have a discussion. Gut health, yeah, present in the gut. The microbiome aids with digestion and immune response, absolutely. Probiotics, yeah, we'll talk about those. Unregulated probiotic market, totally. Vaginal microbiome, yeah. Gut microbiome is the second brain. I'll touch on that as well. Antibiotics, yeah. Great, so I'm gonna give about 30 more seconds. Awesome, mental health and immune system regulation, absolutely. We'll touch on that a little bit. Yeah, our skin microbiome and the gastrointestinal microbiome. Great, I'll get a few more answers. Antibiotics, absolutely. Okay, it looks like we're mostly done answering this one. So let's go back to our presentation. Thank you for your input. So of course, microbiome research has really expanded in the past couple of decades. And there's a huge focus on the human-associated microbiomes. So this figure, I think, does a great job of showing the different symbiotic microbial communities that are associated with human hosts. It's important to know that in addition to the amount of microbes, that's the percentages shown on the slide, different sites can have different composition and diversity of the community as a whole as well. So what I mean by that is some sites might be dominated by only a handful of species, whereas others are going to be dominated by thousands of different species. So the GI tract or gastrointestinal tract is often called the gut microbiome. And this is the site on the human body that harbors the most microbial diversity. And when I say diversity, I mean both in terms of the microbiota or the microorganisms themselves, as well as the microbiome as a whole, which includes any molecules or products that these microbiota are producing in the environment of our gut. So the gut microbiome is increasingly associated with human health and disease, and it's sometimes even called a symbiotic organ due to its ability to impact our health. This is what I did, or I focused on for my graduate research project. And so that's what's going to be at the focus of our chat today. So just a very quick and brief overview of kind of what is known about the gut microbiome. So we know that individual gut microbiomes are established at or right around birth. And there are some major changes when we are developing and aging. So for example, when we start eating solid foods, there's a major change in our gut microbiome. However, they can be relatively stable as a healthy adult. Most importantly, we know that individual microbiomes can be highly variable. So that means different from person to person. We know that the composition of that microbial community can depend on things like genetic factors, geographical location, diet, exercise, smoking status, and we're even starting to understand how factors like pollution and like even having pets might influence our microbiomes. So the real takeaway from this is that, two healthy people can have microbiomes that look quite different. There's no one correct microbiome that we know of now. And this is in contrast to other areas of human health. So let's say you are a cardiologist and you are studying heart disease. So we have a very clear picture of what a healthy human heart looks like, right? And how it's supposed to function. So we can easily identify any deviations from that normal function. But microbial communities are much more complex. So we have our definition of the microbiome as a microbial community, but you can also take this definition literally and consider it as a microscopic ecosystem. So just like the macro ecosystem shown in the background here, our gut contains organisms, and those organisms are in part shaped by their environment. So things like physical barriers, nutrient availability, those are all going to determine where different organisms can survive. And we know that organisms can also interact with and shape their own environment through their activities like resource consumption, waste production, and even competition between organisms. So Apply that thinking to the GI tract, which is an equally complex environment. So the GI tract is dynamic along the length, so moving from the mouth down to the stomach and through the intestines. And although the GI microbiome or microbiota is usually referred to as a single community, the bacterial density actually changes pretty dramatically between the stomach and the large intestine. And this is determined by biochemical gradients that exist along this same axis. So things like pH and oxygen availability are in part responsible for shaping the microbial communities at a given location. As well, the microbes can interact with and influence their own local environment by molecular signaling with host tissues that are co-located with these dense microbial communities. So we have the ENS, which is our enteric nervous system, and as well gut-associated lymphoid or immune tissue. These can receive signals from the microbiota and they can respond accordingly. So it's really a very complex environment to study. So measuring this complex and mixed community can be very difficult because different approaches might actually be biased towards certain organisms. So different cell types, for example. In order to observe the entire microbial community with little bias, we need to go down to those basic building blocks of cellular life. So here we have the central dogma of molecular biology, which, just as a refresher, states that DNA is transcribed into mRNA, which is translated into proteins, and proteins and metabolites are the molecules that carry out the functions of cells. So we can use these different building blocks to study mixed communities, and the approaches vary according to what type of material you're studying. So grouped together, these techniques are called meta-omics. We have things like metagenomics, metaproteomics, you get the idea. So these techniques aim to describe two aspects of microbial communities. Who is there? That's our microbiota. And also, what are they doing? That's our microbiome, right? So for my own study, I use metagenomics and metatranscriptomics, and these are two techniques that are based on the nucleic acids, DNA and mRNA. And that's what I'll be talking about in the next slide. I also want to say that even though microbiome research appears to be predominantly a microbiology field, you might have guessed from that last slide describing the techniques that there's a lot more to this area of science. So for my master's project, I spent a good chunk of time in the lab, but actually most of my work was done on the computer. So let's say you are working on a microbiome-based project. The project is going to begin with a research question, and this research question can come from a variety of different fields. So your research question might be based on immunology. It might be coming from a public health perspective, or it might be part of a genomics project. When it comes to the actual workflow, there are several main steps. These were the ones I used in my project, and they also require mixed skill sets. So on the lab side, you have sample collection and extracting nucleic acids, DNA and RNA, and this requires a mix of microbiology, biochemistry, and basic lab skills. Once you go through DNA or RNA sequencing, from that point on, actually, all of your work is on the computer, and analyzing sequence data like this is called bioinformatics. And this involves a mix of computer science, data science, also statistics, ecological and biology theory. So it's a really, really mixed bag, and it's very interesting to work in a field that has input coming in from so many different angles. I like to think of it kind of like this complex river delta, right? So when we first start out learning about science, we learn under this big umbrella of science. And then when we go to high school, maybe it branches off into some major branches, maybe physics, biology, chemistry, math. If you stay in school and keep learning about science, these topics are going to get more and more granular, right? So math might get broken down into computer science or statistics or many other areas. And I like to think of microbiome research is kind of this like downstream pond where all of these little bits of science are coming together to make their own field. And it combines several of my own interests, which is great. So I have a biochemistry degree, but I've always been super into ecology and natural history. I watch a lot of Planet Earth. And so microbiome research actually allows me to combine these interests. As well, that means that advances in microbiome research have been due to advances in other fields. So we have things like DNA sequencing technology, which can go under the umbrella of chemistry. Computer science, artificial intelligence, and machine learning can be under the umbrella of computer science and math. And of course, microbiology culture techniques under the umbrella of biology, microbiology, and maybe molecular biology. So there are some road bumps or growing pains when it comes to working in a growing multidisciplinary field. So, imagine we have a microbiologist, a computer scientist, and a wildlife biologist, and they're all working on a project together about microbiomes. So you give them a research question, and they're probably going to each come up with a different approach from their respective fields. So which approach is correct? The microbiome field has had this problem. So some early explorations of microbial communities used statistical models from ecology community studies, and they applied these to microbiome sequence data. More recently, data scientists have pointed out why this approach is flawed and it can actually lead to incorrect results. And part of the problem comes from the fact that we often call DNA sequence data, we call them counts. So there are actually key differences between ecological counts and sequence data counts. So an ecologist in the field might observe, let's say, 2 leopards and 60 ladybugs. So these numbers represent true counts. They're observed directly from their environment with minimal interference from the scientist, and there's no upper limit of observations the ecologist can make. So the ecologist is not going to say, I can only have 62 observations. So if I observe one more ladybug, I can't count one of the leopards. In other words, the observations of different species are independent. On the other hand, we have to rely on sequence data to observe microorganisms from a biological sample. We can't just look at them and count them. But sequence read counts are not true counts. So even though we say 50 reads are from E. coli, the reads are actually compositional. So I'm not going to get into this too much to say, but just that it's more like similar to a proportion or a ratio where changing or adding one component is going to affect all of the other components. So the take-home point is that issues can arise when ecological models are applied to sequence data because they'll produce a seemingly informative answer, even though the data are not actually well suited to the model that you're applying. So the answer to our original question is that all of the scientists have to work together to apply concepts from their respective fields in a way that's appropriate for the task at hand. So beware of, copy and paste solutions. A new field really takes time to develop and troubleshoot. So now let's talk about some advances in understanding the gut microbiome and health. And some of you touched on these already in your comments. So, I think public perception of the microbiome is often tied to kind of dietary fads and dietary advice, right? So you've encountered advertising for pre or probiotics, or maybe you've heard advice about which foods to eat in order to have a healthy gut. So that advice is not necessarily wrong, but I don't think it properly represents the aims of microbiome research for human health. What that is like is more nuanced, and we're looking at really particular interactions of the microbiota and their hosts. So you might have a bacterium that someone is studying, and it produces a particular compound, and we are looking into how that compound can interact with our immune cells and what the ultimate outcome of that interaction is. So it's a lot more granular and a lot more specific than, eat this to fix your health. So I'm going to go over some of the things that we've learned from modern microbiome research and a little bit about how we got there first. So a lot of microbiome research is based on something called the hygiene hypothesis. And this states that early life exposures to microbes help to train the development of our immune system as we develop. So this is supported by findings in germ-free animal studies. So germ-free mice, for example, are raised in a completely sterile environment and they don't have a gut microbiome. Now, researchers have found that these sterile mice are prone to developing health issues, and they may be more susceptible to injuries like chronic illness or infection. These negative health effects, or some of these negative health effects, can also be seen in animals that are given high doses of broad spectrum antibiotics. So this can effectively clear out their resident microbiome, causing these negative health effects again. So foundational studies like these have helped us to appreciate really the huge impact that the microbiome can have on human health. And I saw that reflected in some of your comments. So what are people doing with microbiome data? Well, one thing that I read a lot about during my master's project was these folks who are using a type of artificial intelligence called machine learning to classify and even predict the risk of colorectal cancer from someone's microbiome profile. So while we're not really there yet, I just wanted to give an example of the kind of work that is going on and how people are using microbiome data for human health studies. Another application of microbiome research is something called fecal microbiota transplants, or FMTs. So this is just what it sounds like. Essentially, stool from a healthy donor is transferred to a recipient in order to alter their microbiome composition. And I just want to proceed with caution here because this is an area where we do see some sensational results. So the intention of this treatment and what it's been used for is primarily to recolonize the gut of patients that have had a hard to treat C. difficile infection. This often occurs after their own microbiota is wiped out following antibiotics in a hospital setting. So it's had a really high success rate in these patients specifically. But other researchers are looking into FMTs to treat things like inflammatory bowel diseases. So this is like Crohn's and colitis. But I've also seen companies advertising FMT for things like weight loss, or even FMTs from pro athletes that are supposed to help you get really fit. So, always use caution when interpreting results from an emerging field and kind of beware of buzzwords that you might hear. Finally, I do want to give a huge shout out to the Human Microbiome Project. So this was developed a couple of decades ago, and they're really a good place to start if you're interested in microbiome research. One of their aims is developing analysis tools and algorithms and best practices for actually carrying out microbiome research. So some of those meta-omics techniques that we talked about before. The issue is that because microbiomes are compositional, as I said, and highly variable, small alterations in study design can lead to huge differences in the results between studies. So this is what I did for graduate studies, and we actually found that just processing your biological sample in slightly different ways can drastically alter the microbiome profiles that you observe. So this is an example of some real microbiome research and some results. So there is a really interesting cohort study called the Child Study, which is based out of Manitoba. I have nothing to do with them. I just think what they're doing is really interesting. And they're basically researching the symbiotic relationship between humans and microorganisms in order to understand how human development is linked to microbial exposures. So it's becoming increasingly evident, and some of you had this in your comments actually, that early life exposures can impact chronic illnesses later in life. So the Child Study is looking at things like neurodevelopment, allergies, asthma, obesity, and other health issues. I do want to draw your attention to some carefully worded terms. So these are terms you might see in a real scientific study. So you're going to see things like may influence, or we're talking about the risk of an illness and not necessarily, preventing that illness or curing it or getting rid of it. So scientists are usually really careful not to overinflate their results. Instead, a careful scientist is going to describe exactly what was determined from a given study. On the other hand, we have a little more sensational results. So I found these, they're not too out there, but they were, just a quick Google search for probiotics will get you kind of advertising like this. So you can notice on these posters, we have the opposites. We have some broad sweeping claims, right? Fixing your health, transforming your health from the inside out, rebalance your digestive system. So just when you see things like this, make sure to kind of ask yourself, like, what is this really saying? And do we have science that would support results like this? Also, remember, everyone's microbiome can be different. So, how do you think these companies are so confident that their product's going to achieve all of these results for everyone? So we talked about FMTs a little bit, and we talked about altering your microbiome, but there's less evidence when it comes to altering your stable, healthy adult microbiome, or how long those alterations would last, or how they would ultimately impact your lifelong health. So just remember, that individuals have very varied microbiome structures, and what works for one person may not for another. So now that we've learned a little bit about this and we've looked at some more sensational results, I want to know what you guys think about this type of advertising. So do you guys think we know enough about microbiomes to make these kind of claims? I'd like to have just an open discussion for the last couple of minutes here. So you can head back on over to Mentimeter or just go ahead and unmute yourself. I'm going to open up the chat as well. And we can just have a bit of a discussion until we move on to the next portion. Okay, I'm seeing in the chat, what does GALT stands for? So I'm going to type it out as well, but that stands for the gut-associated lymphoid tissue, which is a part of our immune systems. Yeah, this is a good question. So someone's asking, what does the prefix meta refer to in the different types of omics? So I like the way that you phrase this. So you have different types of omics. So you can do genomics, you can do proteomics, but when you apply this to a whole community, it becomes meta-omics. So if I'm just looking at my own genome, it would be genomics. If I'm looking at an entire group of people and all of our genomes together as a community, that's meta-omics. I hope that clears that up. How do we get a regulated market for probiotics? That is way beyond the scope of what I know, but I think, that's a great question, something we should be thinking about. Is there such thing as too much probiotics? I guess it would be possible. I don't know. I think the way that I think about probiotics is kind of the same as vitamins, right? So everyone's different and you're all starting from a different place. So some people are going to benefit more from the addition of those extra metabolites and those organisms than others who maybe don't need them. So you can think of, introducing a new organism into an already established ecosystem. So is that organism going to fit into that ecosystem well? Is it going to last there? Is it going to last there long enough to give you health effects? That might depend on what the starting point of your microbiome is, right? I think that a lot of people kind of look to probiotics if they've just taken a round of antibiotics, or if they've maybe just had a gastrointestinal illness and they're looking to kind of reset. I don't think there's anything wrong with taking probiotics. I just, I'm always cautious when companies are promising these big health results based on probiotics. How do we know what's legit and not when actually interested in prebiotics? So you can do a little bit of research for this. I know like, again, it depends on how things are advertised. So prebiotics are things like fibers and other, basically it's food for your gut microbiome. And so this can come in the form of, fibers, oligosaccharides, it could be vitamins. So, you know, hopefully it wouldn't be something that you couldn't already get in your diet. But I have not surveyed what's out there in terms of prebiotics. I would say, you know, if you can get it from something that you would already eat, then that's probably a safe bet. Yeah, since everyone's microbiome is different, is it safe to advertise a single probiotic to everyone, as some companies might? I think some of the advertising is really based on kind of fear. So, you know, it's thinking if you're not taking this probiotic, you're not up to your optimal health and your optimal gut health. I don't think that's necessarily true, although I think a lot of probiotics that are on the market right now are relatively safe, and they might even be based on bacteria that you already have in your gut. So I don't think they're dangerous, but I don't think it is right to advertise them as some kind of health fix. I'll just look at a couple more questions and then we will move on. I do want to address this one. Are antibiotics dangerous? That is a loaded question. So I think the overuse of antibiotics can be dangerous. Yeah, so that someone has just piped in, how does antimicrobial resistance and the overuse of antibiotics impact the GI microbiome? So this is related to, without spending too much time on it, this is related to that FMT and that C. difficile infection. So if you get a really hard to beat gastrointestinal infection and they can't treat it with antibiotics, you know, you might be a candidate for a fecal microbiota transplant or something like that. So it's hard to describe in a sentence, but that's definitely an active area of research. A lot of people are looking into antimicrobial resistance, overprescribing antibiotics, overuse of disinfectants and things like that. That's all related to that hygiene hypothesis, right? I'm not too sure about specific strains of probiotics. I think the ones that are patented probably just have their own very specific mix. Yeah, so gut microbiome can move outside the GI tract. I'll just answer this one and then we'll move on to the second part. So let's say if you had your gut punctured, certain parts of your body are meant to be sterile. So if bacteria get from your inside of your gut into your abdominal cavity, for example, that is no good. And that's part of what we're talking about with training your immune system. So there are certain places where your immune system might expect to have resident bacteria in the gut. And there are places where those are going to cause an extreme immune response. Okay, awesome. Thank you so much for participating, everyone.